So, introduction to NGS sequencing. So in this lecture, we're going to go right back to the very, very, very beginning to describe what DNA is, why the structured DNA is important for the way that DNA sequencing works, and also explain how specifically Illumina next generation sequencing, although that's sort of an outdated term now, how this data is generated. This may be quite basic for some of you, but if you're like myself and actually has a degree in archaeology and not biology, this might be a good recap to make sure you're up to speed, because a lot of this stuff will be very important for understanding how you process your data later on. So let's start with what is DNA. So probably this thing you're at least roughly familiar with if you're doing a degree in metagenomics. Obviously, we know this very famous double helix thing, which is derived from two strands of different nucleotides, which have a special pairing.
Yeah, I just want to point out that James and I were just in Cambridge for a genomics conference and got to sit in the booth where Watson and Crick were discussing this, and there's a big plaque on the wall. We'll post the picture. Yeah, it's kind of fun.
We can update the slides or something like that.
Yeah. And there's a photo of Rosalind Franklin too.
Yeah. So again, as you're probably mostly familiar, in your DNA you have 4 different nucleotides. These are the pyrimidines, which is cytosine and thymine, and the purines, which is guanine, adenine. You have this base pairing where almost always, or 99% of the cases, you should expect to have one pyrimidine binding with one purine. So whenever you have a C or a cytosine, this will always be on the other strand, G or guanine. I try to remember this as CGI. And then if you have an adenine with A, you'll always have a T on the other strand. Or I remember this Adat Walker from the Star Wars. As you can see, I may be a little bit of a nerd. What this means is that they are complementary. So whenever you find a C on one strand, you should always find a G on the other, and vice versa. And the same with A and T. Then when you want to make a new copy of DNA, aka replication, the way this works is you unwind the DNA sort of primary and secondary structures, you separate the 2 strands out, and then you have DNA polymerases, which basically reads along the exposed area of the single-stranded part of the separated strands. Whenever it finds a C, it will pluck out from the sort of cellular soup the corresponding nucleotide. So in this case, if it's a C, it will put in G, bind that on, and then another polymerase will come along and basically repair the backbone. And this replication concept of separating the strands, finding a new nucleotide, incorporating into a new strand is very, very critical for basically pretty much all of sequencing. So how do we get DNA? Probably, again, particularly come through biology. You're very familiar with this. You take your sample, you lyse the cells to basically break up the cell walls and membranes to basically free the DNA. You then spin it down or something like this to separate the different components out. And then you typically can put out very long, very high quality bits of strands of DNA. If you've done the strawberry experiment, you can actually do this at home and actually see the physical DNA. But this is for modern DNA. Instead, ancient DNA, while the process is the same, the difference is the quality of the molecules. You can imagine your modern DNA molecules as very long bits of spaghetti that you've cooked perfectly. Ancient DNA is when you were to boil it for 3 hours too long, and it becomes all mushy, broken up, squidgy, torn up. It's just a horrible mess. And that is how I like to imagine what ancient DNA is. So ancient DNA molecules are degraded. They are fragmented into very small numbers of base pairs. They're also damaged. So this is something that you'll hear a lot from Tina about tomorrow morning. And then also we have a lot of contamination. So pretty much all ancient DNA samples have a lot of modern contamination around. So imagine you try to hide your really overcooked spaghetti in a new batch, or a lot more, so people don't see. So then sequencing. What is sequencing? Sequencing is basically converting the chemical nucleotides in the molecule to the digital ACTG that you can see in your text editor on your computer. The basic concept in most methods is via replicating a strand. But then when you're adding the new free nucleotide to the exposed one when you're doing this replication, is you add some form of modified nucleotide, typically fluorophore modified, where each of the complementary bases will have a different color attached to their fluorophore. So that means is that each nucleotide will be associated with a particular color. And then what you do, you'll somehow fire some way of getting the fluorophore to emit that color. You record that color, and then you rinse and repeat by adding the next nucleotide and do the same thing. So historically, one of the first, let's say, high throughput, but big bunny is here, techniques is called Sanger sequencing. The way this would work is you could make lots and lots of copies of your molecules, but at different lengths. You would then append onto the end, or actually ligate the end of each of the molecules a different nucleotide, which is modified, but to prevent any additional nucleotides being added onto the end, so blocking nucleotides. You would then run this through a gel to separate out each molecule. So it's all the same sequence, but just lots of copies of the same sequence.
Okay.
Truncated. The gel then separate it out by size, and then you would record each molecule as it goes through and record the light emitted by these blocking nucleotides. As you can imagine with gels, this is very, very slow to pull all of these copies through the gel, separate them out. And while it was definitely much better than what there was used previously to identify what DNA sequences were, this was quite slow. So we won't go into too much detail about this, but this is the basic concept again. As you can see, you're somehow modifying lots of copies of the nucleotide molecules to different lengths, then somehow separating them out in different sizes, and then recording the color in the fluorophore as it goes through. The problem with Sanger sequencing is that it's very slow, it's very expensive, and resource hungry. You need a lot of DNA, and you can only basically do 1 to maybe a few thousand molecules at a time. But in around 2004, 2005, we had this revolution that at the time was called next generation sequencing. It's not really next generation anymore. We've moved on to sort of the third or even fourth generation, but generally most people still call it NGS sequencing. And these techniques allowed you to sequence billions and billions and billions of DNA molecules at once, but also very fast and also very cheap. So at the time, there were a range of different platforms, such as PacBio, IonTorrent, and Illumina, but by far, Illumina has become the most popular because their particular method is most accurate. PacBio very far has caught up. IonTorrent, I don't think really is a competitor anymore. And one important thing is Illumina is what 99.9% of ancient DNA sequences have been generated on. So that is the data you'll almost definitely encounter. The latest generation, maybe third or even fourth generation, is stuff like Oxford Nanopore, which you may have heard of. But we don't cover that for reasons you'll find out later on. NGS works basically the same way as Sanger sequencing, in the sense that you're incorporating a new nucleotide at the end of a molecule, getting the fluorophore to emit a light, and then you basically record each one. You can basically imagine, based on this GIF, this is what you'd be looking at. And you can see just how many different points of this tiny part of this GIF is using. So just scale up by a few hundred times, and you'll just imagine how many DNA molecules you can sequence at once. And the way this works is you get your DNA, and then you want to bind it to what we call a flow cell. So a flow cell is basically a glass slide, where on this slide you have lots of very short synthetic DNA sequences which are bound and immobilized, so they can't move away. You then inject your DNA on, and your DNA molecules will flow over this flow cell, or this lawn of these sort of grass bits of oligonucleotides, and your DNA molecules will bind onto these synthetic DNA sequences to hold them in place. But to get your DNA to be bound in place on these synthetic lawn, as we call it, you first actually need to modify your DNA to convert it into something we call a library. The library consists of adding adapters. These are the things that actually have the complementary sequence to the synthetic sequences on the flow cell and hold them in place. But these adapters also hold priming sites, because this is what you will use with for the polymerases when you start incorporating new nucleotides to basically start from. And also another trick, which is also very, very popular in ancient DNA, is to add another set of synthetic sequences called barcodes or indices. This means that you can actually sequence multiple samples at once, because each molecule of your DNA sample will have a unique sequence which corresponds to that sample, and then you can sort them out after. So this is what an Illumina DNA library construct looks like. So you can imagine this black bit in the middle is your DNA molecule from your sample. You then have primers, which is here in blue and green, on sort of ligated onto the end of these molecules. You then have the sample-specific index. So every sample, or sorry, every molecule of that sample will have the same sequences at both ends, which allows you to separate them out after. And finally, you also have the physical adapter, which also includes another primer sequence. And this adapter is what will is complementary to the oligos on the lawn, so the flow cell. But there's one problem, which is that, as you can imagine, a DNA molecule is very, 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 very small. And a single nucleotide will, with the fluorophore, will not emit much light. So what Illumina has is a process called clustering. And this is a way of making the light emitted by a molecule bright enough to be captured by a camera. And the way this works is, firstly, your DNA will flow over to the... First, your DNA will flow onto the flow cell and bind to one of these synthetic nucleotides on the flow cell lawn. There is gaps or space around each... the person's joined. There's a space around each synthetic oligo on the flow cell, but you also have lots of complementary oligonucleotides very closely around this original one. So it's not just a equal lawn. You have little clusters of these things. So after the DNA is bound onto the flow cell, there's some fancy chemistry which causes the DNA to bend over, and the adapter on the other end of the library construct we saw before will bend over and bind on to another one of these complementary oligo sequences. Then you add a primer and actually synthesize, or I forgot to say, this is a single-stranded DNA molecule. You then bend over the single-stranded molecule, so it's bound at both ends. Then you have the polymerase, which then reconstruct the double-stranded DNA again. So then you have, as you can see here, a double-stranded molecule, both attached to both ends. Then the next step is what you do is you clip off one of each end. And so you basically expose then the forward strand and the reverse strand as single-stranded molecules again. These are basically copies, but complementary. And then you repeat again. So then you'll bend these over, polymerize, synthesize the double strand, cut again both ends, and you do that over and over and over again. So all at the same spot on the flow cell is lots and lots of copies of the same molecule. So it's basically a concentrated area of copies. Yeah. And then you basically cut off one of these ends, so you have all the same sequence in one place. So now in your cluster, so when you have done this clustering process, you're then ready to do the actual imaging of the sequence. And as we said a bit before with the Sanger sequence and things like this, the way this works is firstly on the single-stranded molecule, which is floating in on this lawn, a modified nucleotide will float in, be bound on to the next position. This is also a blocking nucleotide, so we can't allow the next nucleotide to be added on yet. The difference with Illumina sequencing versus Sanger sequencing is that this fluorophore acts as the both the light emitter and the blocker. So you then fire a laser at the molecules, which will emit the particular light. You then have a camera which will take the picture and record the color at that position on the cluster on the flow cell. But then the trick with Illumina sequencing is you cut off the fluorophore. So it's not a blocking like a permanently blocking nucleotide, you can actually reverse this to remove it, and then you can actually add the next nucleotide to be added on. The next in sequence on the single-stranded molecule, this will have a different fluorophore. You then take another, fire a laser, take a picture, cut it off, add the next one, and so on. So you don't have to waste all of your DNA to get the entire sequence. Typically with Illumina sequencing and kits, you have this number of cycles, or basically adding, imaging, cutting off, with either 50, 75, or 125 times. And these are basically called cycles. And again, to revisit what this looks like, if you're imagining you were in the machine, if you were the camera, in the first cycle, you would see light being emitted of two greens. So you can see this is a T and a T. On this one, on the second cycle, you see a blue and a green. You see a blue here and another T here. And it basically repeat. And this is happening. So each of these molecules, each of these little dots on these images are clusters. So lots and lots of copies of the same molecule. But again, imagine this spread out over billions and billions of clusters on the same flow cell that you're having one. So you can see why this is super high throughput. It is not that simple though. With Illumina, there's actually three different methods of actually imaging. We're only going to cover two because these are the two you'll most commonly found. One is four-channel system or four-color chemistry. This on the left. This is what we've just described. There's another method called two-channel system, or two-color chemistry. These two you typically see on Illumina NovaSeqs and Illumina NextSeqs. And instead of having four different colors, you only have two, where green is a T, red is a C. And then if both green and red is emitted at the same time, so the fluorophore actually emits two wavelengths, that's an A. Then if no light is emitted, the machine reads that as a G. There is an artifact about this, which are going to go important later on, which is often something that people forget. So keep that in mind. So there's two different types here. So one issue, though, is this is biology. Biology is messy, and biology gets tired. I am very tired at the moment. This applies to sequencing too. So over time, when you're doing these cycles, the reagents starts getting a bit more dirty. The camera gets slightly out of focus. Basically, the whole system gets not as accurate, and you get more and more errors as you get through each different cycle. This can happen because sometimes the wrong base will bind on into a molecule, or the nucleotide does not actually bind on properly. And sort of over time, the light's being emitted in the cluster gets less clear, so you're mixing different wavelengths. And so the machine will actually calculate a base quality score, which is the probability that it thinks that it got, or the confidence score that it got the right color being emitted from the thing. If it has no idea, it will just report the nucleotide as N. I don't know what was there. This was an issue with Illumina sequencing, and so they came with a clever system to actually correct for this and improve the base quality sequences at the end of the molecules when the machine gets tired. And this is paired end sequencing. Again, quite clever. You remember this bridging amplification when we were flipping over the molecules on the flow cell? Same applies here. So once you've done this clustering, you then sequence one end of the molecule, and you expect over time the confidence or the quality gets poorer. Once you've finished that sequence, you'll then actually flip all of the molecules over, cut them at the other end. So now you have the molecules, but in the reverse way. And then you start, again, the sequencing process again, but from the other end. So where the tired molecules or tired base calls at the end of the first round of sequencing is the base quality is quite low because of the end of the sequences. Now you're starting when the reagents are fresh and the machine is nicely calibrated from the beginning from there. And then what you can do, you can basically overlap the two molecules that you sequenced and then take an average. And that means you can actually recover the base calls which are made at the end of these molecules. And the bonus thing is also, if you do this, is that if your DNA molecule is longer than the number of cycles you have, you actually recover the true ends of the two ends of the molecules you have. So you may be missing the middle bit, but there's clever ways of getting around that. But you have both ends of the molecules, which is very useful. Whereas if you can imagine, let's say your DNA molecule is 200 base pairs long, but your sequencing is only 50 cycles, you'll only get to 50 base pairs of that molecule. You don't, it's the other end. You don't know when it ends, or you don't know how long it is. But if you sequence the other end, you actually recover this other end. So you've now had the machine cycle through the different bases of each molecule, and you've recorded all of the lights and being emitted. How do you actually convert this to computational sequences you can do analysis on the computer? So the machine, like Illumina HiSeq or Illumina NextSeq, will do the actual conversion with a little server of the light to an actual the ACTGs in your file. But the first thing you need to do is also at the same time is separate out all of the molecules into each of their index sequences. So remember I mentioned these adapters and the barcodes. You need to separate this out so you know that this molecule comes from, these sets of molecules come from this sample. So this process of converting from the light to the ACTGs on your file, and also separating out into different files per sample, is happen by demultiplexing. So basically you can see here where you have all of the different colors. Each color combination, like red, green, red, yellow, these represent two distinct sample. And so demultiplexing pushes these into one sample, which has just the red and yellow indices, blue and yellow in this case, and so on. And the output of this demultiplexing process is your FASTQ file. And your FASTQ file, according to Wikipedia, but is actually probably more accurate than AI nowadays, is that FASTQ is a text-based format for recording a biological sequence, typically DNA, but also with a corresponding quality score. So when I mentioned about the machine trying to get an estimate how confident it is that it's got the right nucleotide, this is also stored in the FASTQ file as well. And both this sequence and the quality score is encoded with single ASCII characters, which you can see here. So FASTQ files are text. They are not some fancy binary format that you have to load in fancy programs. It is just text. You can't always just read them straight off. And what you would see when you look into it is a repeated set of 4 lines. The first line is this, which is a read ID. Then you have the sequence. You have a spacer gap. And then the quality scores encoding this weird random set of letters and numbers and characters. So the way this works is that in the read ID, typically when it comes off the sequencer, people do modify it sometimes, you will have the ID number of the sequencer itself. Then you'll have a flow cell ID, which is actually the actual like ID of the flow cell, the glass slide that you had in the beginning. Then you'll have coordinate system to tell you where on that particular flow cell the cluster that this molecule has come from is. And then often at the end, you'll have information about the indices or barcodes that it was demultiplexed with. Then you have the DNA sequence. You can see here you have A, C, T, Gs, blah, blah, blah. But you may also notice right at the beginning you have this N. Does anyone in the room here remember what the N stands for? Unknown, exactly. So the machine did not know what color was at that position. So it has recorded this as an N. And then you have the spacer, which basically does not do anything anymore. So it's just a space to make sure you have lines of 4, which is nice for dividing. And then you have these random characters, which corresponds to this quality score matrix down here. The actual characters and the number it corresponds to does unfortunately depend on the platform you use. So this is not going to be the same for everything, but you get the rough idea. But basically, if you see an exclamation mark in most Illumina sequencing, that means you have a base quality of 0, or even in this case with our badly called base right at the beginning, which is actually when the camera is calibrating on the first base. That is a hash, which is very low quality. In contrast, most of the molecules you can see here, most of the nucleotides have got quality scores of J in this one, which corresponds to 41, which is the highest quality score. So it's a very good quality base score. And pretty much all of bioinformatics, genomics will start from this raw sequencing file format. So to very quickly recap, we have DNA molecules, which essentially just made up of 4 nucleotides, A, C, T, G. You have the two complementary base pairs, which you then, when you separate out, you basically have the reverse complement of each. And replication is very important because this is where you basically, on the new strand, put in these free nucleotides to reconstruct the complementary bases on the other strand. Modern DNA is very long and spaghetti-like. They're good, sorry, tasty and well-cooked, spaghetti-like. Ancient DNA is very short and crappy, which I think I do come to later on. For NGS sequencing, you are massively multiplexing this, so you're sequencing millions and millions of different DNA molecules at once. You can also do that with different samples mixed all up at the same time, because you can add these adapters, which you also have these barcodes, and the adapters are what allows you to bind to the glass flow cell. So once the molecules are immobilized, then you basically you separate it into single strands, make lots of copies to get the clustering. Then you do basically this sequence of adding a modified nucleotide, firing a laser, emitting, taking a picture, clipping off the fluorophore, adding the next one, repeat ad nauseum. Remember that the desyncing of these clusters, because biology is messy and gets tired, you get the lower base quality scores, but you can also improve this by paired end sequencing. And both the DNA sequencing, the location on the flow cell, and the sequence and the confidence the machine has made to of the base quality score can be improved by this paired end sequencing, and is recorded in the FASTQ file. Right, now we're getting into the more nitty-gritty, more sort of complicated things why ancient DNA is a bit more special. So the next things, particularly if you work on ancient microbes or ancient metagenomics, but also if you work on ancient popgen, you probably want to take these things into consideration. So the first thing is that ancient DNA is often a very small proportion of all the DNA in a sample. That is, because the DNA is not very well preserved because biology, it gets eaten by microbes, it gets attacked by water and chemistry in the soil and things like this. And what this means is that many, many ancient DNA researchers have to amplify many times prior to sequencing to get a sufficient amount of the molecules so that you can detect it in the sequencing machine. This means that you can inflate counts, so the number of molecules you have, but also you start getting duplicates where you just sequenced the same DNA molecule multiple times because you're doing this amplification. You have to be careful with over-amplifying because this can actually reduce the number of sequencing slots that your machine has available. So you imagine this as the number of clusters your flow cell has. And if you have too many of these duplicates, then you're basically wasting money because you're not getting any more information because you're sequencing the same molecule over and over and over and over again. So this is something you should always check. Another thing is something called index hopping. This is particularly if you're getting data from Illumina sequences that use patterned flow cells. This is a recent technology that's been added to HiSeq X and NovaSeqs. The patterned flow cell are basically this glass flow cell, like we mentioned before, but instead of being randomly distributed, they actually have tiny little wells. But they have this slight artifact that when you try to exclude other molecules going to these tiny little wells, there's a bit of chemistry, which means that if you have free floating index primers, so primers that have not been, or adapters that have not been properly bound onto your molecule, they'll start being added onto different molecules from different samples. As you imagine, that's quite bad, because then you start getting molecules from one sample being assigned to a different sample. And so this is something, if you're getting data from the NovaSeq or HiSeq X, you should try and check for. There are different methods of doing so, quite a few papers on this, so check this if you imagine this. In particular, you should be careful about things such as if you are doing capture or better example. We had lots of problems in ancient DNA where we were trying to sequence lots of ancient DNA samples, and suddenly we had lots of ancient COVID or SARS-CoV-2 genomes because there was lots of high quality genomes being sequenced on the same sequencing run, because index hopping, molecules from the SARS-CoV-2 sequencing libraries were being assigned or accidentally being given these adaptive sequences in the ancient samples. So we know that SARS-CoV-2 is a more recent thing, so this is something to be careful of. Another thing is sequencing errors. As bioinformatics is getting more and more throughput, people are increasingly more actually forgetting to go check the raw data when it is sequenced. So always when you get sequencing data, always either your core facility or you, but ideally you, to check that you don't have a huge drop off of the number of quality scores at the end of molecules. Because if you do have these low quality bases and you don't clean them up, you risk the problem of sort of incorporating false mutations in your sequence analyses. It can break things like de novo assembly when you want to do overlaps and things like this. So do actually always go check your raw data, and don't necessarily trust core facilities or other people to do it for you. Another thing is that you should always remove your adapter sequences. There's lots of different tools that do this. So I'm not going to tell you how to do it necessarily, but most pipelines will also have a tool which basically clips off or removes the adapter sequences off the end of your molecules when you do the first step of your analysis. This is very important because a lot of people forget to do this, most famously in a carp genome, which means that whenever you have any adapter sequences in your own sample, left over for whatever reason, you will start finding carp in every single one of your libraries. And so particularly if you're doing metagenomics and you're trying to identify, I don't know, what people were eating, you might get excited. Oh, we can find direct evidence that Neanderthals were eating carp 100,000 years ago. Actually, it's just this genome is a dirty genome, which is unfortunately uploaded and not being removed, just has adapters in it. So your adapter sequences in your library are being assigned to the adapter sequences in the carp genome, not actual carp DNA going to carp DNA. That's very, very important, very, very important. Another thing is poly-G tails. So you remember from before I had mentioned that we have this two-color chemistry where you have red, green, both, or nothing. So if there's nothing, the machine assumes that this is a G. What the issue that we have in ancient DNA is often our molecules, as we hear from Tina tomorrow, are very short and actually less than the number of cycles in the thing. So let's say you got a 50 base pair cycle sequencing chemistry kit. Our read length is only 20. You'll have 30 cycles of nothing being emitted because there is no DNA to be emitted. And that means you're going to have lots of G's right at the end. And if you don't remove this, it's going to make it harder for your DNA aligner later on to compare to reference genomes, because it cannot find a place where you have your sequence and then G's at the end of it, and you'll lose a lot of things. Also in taxonomic classification, if it does not align specifically there, you have long tails of G's or G-only sequences. The classifier will not know where to put it, so it will push it up your taxonomic tree and just assign it to bacteria, like the root of the tree, which is also not very useful. So that's the same thing. Okay, so to recap, I think we're 5 minutes behind. Apologies about that. When you get to the analysis, and we will recap lots of these things throughout the summer school, so don't worry too much. But just things to check for, to have on a checklist when you're analyzing data, is check your duplication rate. So that may indicate you have someone who has done the analysis of, wet lab work has over amplified the libraries. Be careful of index hopping. Always check that something like a pathogen has not been sequenced from another library is also present in the same run, and you start seeing it in your own libraries. Check for sequencing error. This is very important. People are forgetting to do that nowadays. Always remove your adapters, otherwise you'll get lots of carp in your analyses. And also check and remove, ideally, low sequence reads, like with poly-G tails that you may get from NovaSeq or NextSeq and NovaSeq. So two-color chemistry things.